Hello, my name is Allyson Kritzer, and I am a second-year PhD student at Cornell University in the Biological and Environmental Engineering Department. Today, I'd like to share my work titled Role of Dechlorinating Microbial Communities in Chlorinated Ethene Back Diffusion from Low Permeability Media. This work is funded by the Strategic Environmental Research and Development Program, or SERDIP, project number ER20-1079. Groundwater contamination from chlorinated solvents like trichloroethene or TCE remains a persistent challenge at many sites. Even after the source is removed, TCE can persist because it diffuses into low permeability zones where it is stored and slowly released back into flowing groundwater over time. Bioremediation through microbial injection into an aquifer promotes reductive dechlorination of TCE to more soluble daughter products such as cis-1,2-dichloroethene, vinyl chloride, and finally ethene. This transformation enhances concentration gradients, driving back diffusion from low permeability zones while simultaneously reducing contaminant toxicity down gradient. The objective of this work was to assess how subsurface heterogeneity and bioenhanced diffusion from low permeability zones control contaminant release and detoxification following simulated source zone remediation. A 64 by 38 centimeter aquifer cell was constructed with a low permeability Martin soil lens. Flow is driven by a constant head difference between the influent reservoir and the effluent outlet. Samples were collected from ports throughout the system over the duration of the experiment. A sodium bromide tracer test was first conducted to characterize the flow field. TC was then introduced to simulate mass storage in the low permeability lens under both abiotic and biotic conditions. The biotic system was bioaugmented with a commercially available dechlorinating consortium. TC and daughter products were measured using gas chromatography. Results of this experiment indicate that bioremediation accelerates TC removal and drives the system toward ethene-dominated end products. However, back diffusion from the low permeability lens still controls long-term contaminant release and persistence. These results highlight that even under active biodegradation, sustained mass transfer from low permeability zones remains a dominant control on plume persistence. As a result, effective in situ remediation strategies must explicitly account for diffusion-limited mass release when evaluating treatment performance and predicting long-term cleanup timelines. Looking ahead, future work will focus on qPCR to characterize the spatial distribution of key dechlorinating microorganisms and link that distribution to bioenhanced back diffusion with the goal of improving predictions of long-term contaminant release. In summary, this work demonstrates that while bioremediation is effective at transforming TCE and accelerating mass removal, long-term plume behavior is still governed by diffusion-limited mass transfer from low permeability zones. Improving our understanding of how microbial processes interact with subsurface heterogeneity is critical for developing more effective and realistic remediation strategies. Thank you.